Okay, so I'm gonna make a little video here and kind of explain DNA replication at HL level for anybody who's still confused about this. So remember the basic problem here is that DNA is anti-parallel. Okay, so one strand runs one way, one runs the other way. So what that means then is um, well, a couple of problems. One is the DNA polymerases only work on adding things to a 3' end. You could also say they only work from going from 5' to 3', right? Um, so that creates complications when the strands are anti-parallel. So looking here, like for this one, notice this says 5', which means this side here is 3'. So looking at the other one, that's 5' to 3' that way. Um, so DNA polymerase 3, for example, can only go on this particular DNA molecule. It can only move that way. Okay, now that's no problem because helicase right here is busy unzipping the DNA molecule and it's moving in this direction. Okay, so as it moves, it's unzipping this, and DNA polymerase is it's actually attached to it. It's right behind it, adding nucleotides in a 5' to 3' direction. It's perfectly happy doing that. No problem on the leading strand here. It's just straightforward. Now, the lagging strand is a problem because here, right, we said that's the direction the DNA polymerase moves. On the lagging strand, DNA polymerase moves this way. Okay, so it's moving this way. Now, the problem is, as it's moving this way, helicase is moving this way, so you're leaving gaps here. Okay, um, you don't have the same gaps as you would have here with DNA polymerase 3 because it's moving in the same direction. Okay, so what that means is DNA replication on the lagging strand is a little bit more complicated. So what has to happen first is right here, we have another enzyme called primase, and it adds a RNA primer. So a piece of RNA that basically is a sign map for DNA polymerase 3. It tells DNA polymerase 3, attach right here and start copying. So as helicase zips down the DNA molecule, primase is right behind it, adding primers. Okay, so you can see the primers that were added here, 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 and here. Okay, so it's leaving little primers. So then what DNA polymerase 3 does, starts at that primer and going in the 5' to 3' direction, it adds some nucleotides. And it adds them up to when it gets to the next primer piece here. Okay? Um, Now, DNA polymerase 3, like other enzymes, is pretty much a one-trick pony. It does one thing. And all this does is it adds nucleotides to the 3' end. So when it gets to this primase section, or to this next primer, it just stops, it falls off, it stops working. So you still end up with a gap here. So what you end up with over that molecule is you end up with gaps. And at the end of each of those gaps, sorry, not that way. At the end of those gaps is RNA primer. Okay. Now we call these Okazaki fragments. Okay, so you see them better up here. So these are the Okazaki fragments. So right here, here, and here. Um, so then you basically have gaps in DNA. Gaps in DNA is generally frowned upon. So there's two other um, molecules involved. So one is DNA polymerase 1. And what it does is it basically cuts, oops, it basically cuts out that, that primer, that RNA, and it puts regular DNA in it, okay? But again, just like polymerase 3, it's a one-trick pony. It only does one thing, and I just showed you what it did, okay? So the final piece for this side is something called DNA ligase. Now, ligase is basically kind of glue. It just joins those pieces together so that now, as I kind of shown here, you get a continuous piece of DNA. And that continuous piece of DNA is an exact copy of the lagging strand. Okay, so the leading strand tends to go a little bit faster, and we call this one the lagging strand because it's got to work backwards, so it goes a little bit slower. So I hope that explains how DNA replication a bit clearer. Really, what you need to be able to do is describe these steps, and you got to be able to describe the role of these enzymes in DNA replication, or what they actually do. Okay, so thank you.